Hi, I'm Dr. T and welcome to my office. So today I would like to talk a bit about the wonderful world of proteins. Proteins are biopolymers. These are going to be polymers, so large molecules made up of many subunits, and they are typically made by living things, hence the bio part of the biopolymer. Now, like all polymers, they are made up of small subunits known as monomers. The monomers for proteins are known as amino acids, and they are going to have a few things in common. Amino acids are going to have a central carbon, what we call the alpha carbon. On this carbon is going to be attached three different functional groups. Well, arguably four. One of which is going to be a hydrogen, but that's not really a functional group, it's just a hydrogen. By definition, that carbon needs to have attached an amine, or a nitrogen functional group, and an acid functional group. This is another carbon that contains a double bonded oxygen, as well as a single bonded oxygen, which is itself attached to a hydrogen. That hydrogen can come and go, hence the name acid. Now, as I mentioned, there's another one. We usually describe this as the R group. R in chemistry simply just means other, kind of in the ellipse of the chemical world. There are approximately 20 some odd amino acids, depending on exactly what you want to call a different amino acid versus a modified, depending on how you want to split it. And some are more common than others. So if you want to be very broad, you can get up into the 30s. But there's 20 standard amino acids, and then there's modifications. These amino acids are different based on what R group they have. They can be as simple as another hydrogen, or as complex as an aromatic ring or two. Now, these amino acids can be stitched together to form either oligomers, which we call peptides, or larger structures in the polymer form that we call proteins. These proteins can come in a couple of different sizes and shapes. They can be kind of a glob shaped, typically with hydrophilic amino acids, ones that like water on the outside. These are what we call globular proteins. Alternatively, they could be a similar shape or somewhat different, but with hydrophobic amino acids on the outside, and ones that would like to embed themselves in cell membranes. We call these membrane proteins because we are not original when it comes to naming. Of course, they don't have to be kind of glob in shape. They could be long and thin, kind of fibrous, to which we call them fibrous proteins. Typically speaking, fibrous proteins are used for structure and support within cells, or without of cells, what's known as the extracellular matrix, the area between cells. This can have many fibrous proteins, such as collagen. Globular proteins can serve several functions, things including transport, like we see with serum albumin or with hemoglobin. Alternatively, they can actually do chemistry and become what we know as an enzyme. Enzymes are proteins, but they are also catalysts. They're able to facilitate the chemical reaction, one specific chemical reaction, per each individual enzyme, and thus help facilitate the functions of the cell. Membrane proteins can be things like transport proteins as well as enzymes. They're a very diverse group of proteins that are as well quite essential to the cell. Now, when it comes to cells' manufacture of proteins, they're going to need the constituent amino acids. For humans, some amino acids are capable of being synthesized, however, others are not. These are ones that are typically relatively complex and would already be present in our diets. These are what we refer to as essential amino acids. They're essential because you need to eat them since you can't make them. Also in the essential category is that some proteins, actually quite a few of them, will have other constituent parts that are not amino acids. Things like metal ions embedded within them or organic molecules. We call these cofactors. Now, some of those cofactors you can manufacture just fine within your cells. Others you can't. Typically, the cofactors that you can't manufacture and require to get from your diet, if they are organic in nature, are known as vitamins. The first vitamins discovered, the B vitamins, because the second vitamins discovered had the better marketing campaign and were called A vitamins, were amines, thus the amine in the vitamin, and they were necessary. They were thought to have vital force. This was right as this idea was slowly dying. Now, these are necessary for human consumption. Typically in a modern day, we do get enough vitamins. If it is not an organic molecule, but an inorganic molecule, or I should say an ion of some sort, usually metal ions, these would typically be referred to as minerals. Once again, you do need to have these, but they're kind of like a screwdriver. You need one when you need one, but you don't need 50 billion of them. More isn't necessarily better, and in some cases can cause problems. Well, that's the gist of proteins. Except I did forget to add something to this video. So while editing, I noticed that, hey, I forgot to talk about denaturation. So proteins, in order for them to perform their function, they need to fold, rearrange themselves into the proper shape, kind of like molecular origami. Now, if they're not in the right shape, they are not going to perform their necessary task. So anytime we break that shape apart, if we simply unfold them or we scramble the folds, that's called denaturation. And that will usually permanently, but sometimes only temporarily, destroy the protein's function. The protein is still there, the amino acids are still hooked to each other, but it doesn't work anymore because it's the wrong shape. You know, imagine if your car got into an accident. It's still your car, it's just no longer the right shape and it doesn't drive anymore. Now, this is actually quite important for us since there's times where we want to break proteins. Say the proteins in some salmonella that's in your chicken. You don't want those proteins working because you want the salmonella nice and dead. So how do you kill the salmonella in your chicken? You cook it. In that case, we're using heat to stress the intermolecular forces that are holding together the protein, and in doing so, it unfolds and is no longer functioning as it was. And hence, the reason you cook chicken is to denature the proteins in any salmonella or other junk in your chicken. Not to mention, it tastes better. Now, you can use heat, you can use a couple of other approaches. Acid is popular, a dish called ceviche, which is popular in Spanish-speaking cultures. Absolutely delicious, that's an acid-cooked fish. And there are other ways of denaturing proteins, such as with organic solvents. But with that said, next biomolecule in the next video. Have a wonderful day.